Hello everyone, in this video I'll be explaining on the introduction of electron microscope. This electron microscope is also an optical microscope, which is a technique for obtaining high resolution images of biological and non-biological specimens. In electron microscopy, a beam of electron is employed instead of the beam of light used in optical microscope. The working of this electron microscope is focused by circular electromagnets, which are analogous to the East lenses of light microscope. Shadow casting is an important technique in electron microscopy. It is achieved by depositing a thin layer of metal on the object. Such metal-coated object held in the path of the beam scatters the electron and produces an image which is focused on a fluorescent viewing screen. This electron microscopy is especially used for rapid detection of viruses in clinical samples and particularly useful for detecting non-cultivable viruses. It is used in biomedical research to investigate the detailed structures of tissues, cells, organelles, and macromolecular complexes. Ultrastructural study of various microorganisms may also be done by this method. There are two types of electron microscopy, which are scanning electron microscopy and transmission electron microscopy. The scanning electron microscope is a type of electron microscope that produces images of a sample by scanning the surface with a focused beam of electrons. The electrons interact with atoms in the sample, producing various signals that contain information about the surface topography and composition of the sample. The scanning electron microscope is a recent development which provides a higher resolution and three-dimensional image of the electron. Specimens are observed in high vacuum in conventional scanning electron microscope or in low vacuum or wet condition in variable pressure and environmental scanning electron microscope and at a wide range of cryogenic or elevated temperatures with specialized instruments. So there is an introduction of electron microscope, where electron microscope has two types such as scanning electron microscope and transmission electron microscope.